Welcome everyone to my talk on modeling fragment counts and improve single-cell ATAC-seq analysis. I'm Laura. I'm a third-year PhD student in the groups of Gina Gagnon and Fabian Theis in Munich. So why are we interested in looking into single-cell ATAC-seq or ATAC-seq in general? So it's a way to look into gene regulation. So most of you know that there are certain elements in the genome that encode so-called cis-regulatory elements, which are elements that bind the so-called trans factors, which could be transcription factors, for example. And then once this is bound, it triggers some regulatory process, like the start of transcription, for example. And this process can be regulated on different levels. So, for example, the binding site could be inaccessible, which could be measured with single-cell ATAC-seq, or accessible. The trans factor could not be available, or there could be certain cofactors that are missing, which would all inhibit this interaction. So in our concrete case of measuring chromatin accessibility, we are looking into open regions in the chromatin, and it has been found that they tend to mark active cis-regulatory regions, so enhancers and promoters. So this is why we are really interested in looking or finding these regions in a given cell. But concretely, what we were looking at in the preprint that I'm presenting today was the different steps of analyzing single-cell ATAC-seq data. So you might have heard that, single-cell readouts in general, but single-cell ATAC-seq data especially are very, very sparse. So you don't get many reads per cell. And the feature space that you get is that for each cell and each peak, you get fragments aligning to these regions, you count them, and then you do further analysis on this cell times peak matrix. And there are other strategies like binning your genome, but if for the purpose of this talk, I won't go very deep into this. And concretely, we wanted to look into the binarization of this matrix, because as I mentioned, single-cell ATAC-seq data is very sparse. So a common strategy that has been employed is that you binarize these count matrices into a 1-0 matrix, with the idea that then you reduce the problem to, is my region accessible? Does it have a read aligned to it, or is it not accessible, or was there just no read captured? So this is very, very often employed in single-cell ATAC-seq data, and we looked into if this is actually a good idea, or if we could improve the modeling by using the actual count data, and if there's also quantitative signal in our single-cell ATAC-seq data. So the way we performed this is that we took our cell times peak matrix, either the binarized version or the non-binarized version, and we employed latent representation learning. In this concrete case, this was a variational autoencoder, and then we modeled the data either in the binary case as a Bernoulli distribution of the latent dimension ZC here, the overall count, the LC, and then some bias factor for each region. It's not very important what these all were, but just for you as an intuition. And the second case was that we actually modeled the count data with a count distribution. In our case, we chose the Poisson distribution. And then we evaluated this using different downstream tests. So the first one was reconstruction. How well do we actually reconstruct our input data using these different latent learning representations? The second one was, I termed here integration, but it was basically how well do we capture different cell states. Also in data that has batch effects, are we maybe better by using the count data? And the third one was that we actually wanted to look into the quantitative signal that is contained in single-cell ATAC-seq data. And to just as a recap how the protocol works, so we have a our chromatin with open accessible regions, and the protocol works using this TN5 transposase, which is an enzyme that inserts into the genome and cuts. It only inserts in accessible regions and cuts the genome there, and then inserts sequencing adapters. And yeah, every pair of transposes, as here in the example, creates one fragment of DNA that can be amplified, sequenced, and then aligned back to the genome to actually find the regions that were accessible. So here I'm just showing an example of the fragment that was generated here, and it's common that this is sequenced paired n, which will become more important later. So you get for each fragment, you get basically the 2 reads that you align. And then, this is for every cell, but then we need to define regions in the genome of interest. So one strategy is to use spinning. The other strategy is to actually call peaks on all the cells or on a subset. And then once we have this region, we count for each cell how many fragments or reads overlap with this. And I want to highlight that this is actually a difference if you call fragments or reads. So in the 10x, which is a common analysis and also experiment framework for single cell, they actually count the read ends with the logic that this should be the insertion site. So in this example, this peak here would get a read count of 1, 2, 3 reads. Whereas if you count the actual fragments, you would get here a count of 2, because this read here is missing. And this actually makes a difference. If you look into the distribution of the count data. So here I'm showing for one of the data sets that we looked at, which is generated using the 10x platform. If we employ the read count strategy, we see this weird distribution where you get more even than uneven counts. Whereas if we look at the fragment counts, we don't see this. And this is just an artifact of the counting strategy, because it's more of a counting strategy because since this is paired and reads, if you find one read, it's very common that you find the second read also in the same region. So normally you will find even pairs of reads, so you get this weird distribution. So the first thing that we argue is that it's to model the quantitative data, we should look into the fragment counts, which have like this like a Poisson distribution rather than the read counts. So going into the first evaluation that we did, that was the reconstruction of our data. And since we had some methods that worked with binary data and some with count data, we had to find a way of comparing the two. So what we did is that we converted the Poisson rate parameter into a Bernoulli probability of observing more than 0 fragments, so 1 or more fragments, which is this typical binarization threshold. And then using this probability matrix, we could compare it to the true binary profile using common classification metrics, and in our case we use the average precision. And just to give you an intuition of what this means, if you're not too much into the maths, if you want to predict the outcome of a football game, for example, you could try to predict team A wins or team B wins, which would be the binary case, or you could try to predict how many goals each team scores. So if team A scores 3 goals, team B scores 2 goals, you could try to predict this and then binarize it later by saying, okay, if you scored more than team A, you won. So this is exactly what we do here. We try to predict the counts and then binarize it later. And this is the outcome for the 4 different data sets that we used for this benchmark. So 3 of them are actually 10x data sets from different organisms and different tissues. And the fourth one is a ATAC-seq 3 dataset, which is multiplex indexing, which is known to be sparser than this 10x readout. And we saw that for the 10x datasets, there is actually a significant advantage in using a Poisson or the quantitative data compared to the binary data. And here the difference between those two is just the way they include the total read count. And yeah, we didn't see this advantage for the ATAC-seq 3 dataset. And we did more analysis in the paper and saw that basically the advantage of using a quantitative model increases the less sparse your data gets, which is also kind of intuitive. So here we argue that the ATAC-seq 3 dataset is most likely too sparse to see an effect. And then the second evaluation, we looked into how well do these different methods capture the different cell states. So this is here an example of a UMAP of the learned latent space of the quantitative model on the NeurIPS data set. And we measured, or we employed different metrics that were introduced here in this benchmarking paper, and they basically try to evaluate 2 different aspects of data of single cell data integration and latent space learning. The first one is metrics that fall into the category bioconservation, and they try to measure how well do we actually capture different cell types if they come from different batches, and also how well do we capture rare cell types that are not present in many batches. So this is the first class of metrics. The second class of metrics is they measure batch correction. So if we have different cell labels, but from different batches, ideally we want them to integrate well after this latent space learning. So this is the second type of metrics that we looked at. If you want to see a very detailed description of the metrics, you can either read this or also read the preprint. So here I'm showing the overall score, which is a weighted average of the bioconservation and the batch correction metrics for, again, the 4 different data sets. And we see that for the 10x data sets, the count-based modeling shows better batch correction and better bioconservation compared to the other methods. So the other methods are very common single cell ATAC-seq dimensionality reduction methods like cis-topic, which is latent directly allocation, or SINYAC, which uses latent semantic indexing, LSI, which are very common dimensionality reduction techniques. Yes, and then we went to the third step that we actually wanted to see what kind of regions have high counts, are these interesting regulatory regions. So the first thing that we did is that we looked into the length of these regions. So we saw that longer peaks tend to, high count peaks tend to be broader, so larger accessible regions than peaks with less counts. And then interestingly, we overlapped these high count peaks, or we did enrichment analysis of these high count peaks with interesting regulatory regions like super enhancers, promoters, distal regulatory elements. And we saw that the high count peaks, they are enriched for promoters of highly variable genes, which are genes that are interesting for this data set, and also highly expressed genes, and also super enhancers. So these are regions, promoters that tend to be highly transcribed and therefore more accessible. Then we also looked into if this accessibility measurement, what we saw before, if a region or a promoter that has a higher accessibility also has a higher gene expression. This is kind of what people also saw in bulk before. So here I'm showing one example for the SLC4A1 gene, which is a gene that is important in this erythrocyte lineage. Here we saw that this is indeed the case, that the higher the accessibility of the promoter, the higher is the expression of this promoter. So this is another indication that single-cell ATAC-seq does in fact capture quantitative data. And then in the last step, we wanted to compare if a quantitative model is able to capture these small quantitative differences between the accessibility of a promoter to better distinguish cell types. So here's again the example of this SLC4A1 promoter, and on the x-axis I plotted the normalized accessibility, which is basically the accessibility that the model predicts for the binary case and also the cystopic case. And then on the y-axis is the expression of this promoter. Again, we see this correlation, but more importantly, we see that the Poisson model is better able to distinguish these three different cell types, or this lineage for this SLC4A1 gene compared to the two binary methods. And we measured this using the silhouette score, which basically is a measurement of how much do these distributions overlap. And with this, I hope I could convince you that we should model the quantitative data for single-cell ATAC-seq, and that there is actually interesting information contained in it. And I also invite you to read the preprint. And I would really like to thank my both labs, so Gia Garnier and Fabian Theis, and then all the people that worked with me on this project and gave me very valuable feedback. So thanks a lot.